All right, thank you all, and thanks to the organizers for giving me the opportunity to speak here today. Just as a brief overview of why we're doing what we're doing, my lab is generally interested in the problem of predicting the effects of genetic variants on gene expression with the aim of developing personalized therapeutics to treat and prevent disease. And so we actually do a large variety of both experimental and computational things. I'm only really going to talk about the one arm of the lab, the machine learning arm, today. So we know that genetic variation can alter TF binding and expression and therefore impact disease. And the way we think this works is by the variant altering the affinity for one or more transcription factors, which changes the binding to the DNA, and that changes in gene expression. And while we have a pretty good conceptual understanding of the way this works, we really do not have the quantitative understanding needed to predict these variant effects in a meaningful way for stratification of patients and predicting disease outcomes. So today I'm going to talk about our work trying to improve these cis-regulatory models so that we can actually accomplish these goals. So a lot of the current AI revolution that we're in with like DALL-E and ChatGPT and all these models has been driven actually by these standard benchmarks where people have agreed that it's a good benchmark data set to use. And so people continually develop new and improved models and test them against these same benchmark data sets, some of which are shown here for different machine learning fields. But you can see that they improve over time and eventually surpass human performance on these tasks. So with this goal in mind, only for cis-regulatory models, we had this dream challenge set up. And we are very grateful to a very large number of people who participated in the challenge, which are shown here, but there's not going to be time for you to see all the names. And in my group, this was really driven by a very talented PhD student, Rafi, in the group. We're also very thankful to our sponsors. We got free TRC time from Google Research, which we were very grateful for kind of leveling the playing field from the competitors, and also Deep Genomics sponsored the winners of the competition to go to RSG Dream in 2022 and present their results. So the Dream Challenge worked like this. There was a very large data set that we had experimentally measured in yeast of promoter sequences. These are random promoter sequences and the experimentally determined expression levels. The reason why random DNA actually works in this context is important, but kind of beyond the scope of today's talk. So if you want to learn more about that, you can look here, but it turns out that random DNA is a pretty ideal machine learning data set for this kind of thing. So over the course of summer 2022, over 8 weeks, the competitors used this data to create increasingly good models, and they had a leaderboard that they were competing to be the top of. And at the end of the challenge, we evaluated their models on a variety of tasks in terms of predicting promoter expression, including things like predicting the effects of genetic variation in the promoter DNA sequences. Over the course of the competition, the competitors' models improved substantially, and by the end, we had state-of-the-art models, where here I'm showing the previous best, this reference model here. This is basically the starting point that the competitors had to work with, and they had to improve on this, although they used a very large variety of approaches. And in the end, the competition was won by this group, autosome.org. I won't talk in detail about their solution, but you can see that across all of the evaluation metrics, they performed very well. And importantly, these evaluation metrics don't all agree with each other. So if you just look at the correspondence between the columns here, the different evaluation metrics revealed different strengths and weaknesses of the different solutions people had come up with. So most of the top submissions, all of the top 10, were neural networks. And it was an open question as to why the winner had actually won, because so many differences existed between all of the solutions. We really weren't sure what it was that made the difference. So we came up with this prefix framework where it's like a menu of options where you modularize each of the models into these different sections that are logically equivalent, although implemented in very different ways. And you can pick one of each item, put them all together, and test that combination to see how good the combination is. Now, it turns out that the solutions here were quite complex in that some of them depended on each other. So in most cases, there was not a clear winner in terms of which module was best, with the exception of the data processor and trainer, where Autosome.org's was clearly superior to the others. And their insight was modeling the data as it had been generated. In this case, the experimentally determined values were based on this cell sorting of these yeast cells by gene expression bins in a cell sorter. And they modeled gene expression as this probability distribution over the bins. And that appeared to make a key difference in why they won. Also, using this prefix approach, we were able to improve all of the individual models. So we kept the core for each of them and changed the parts around it. In some cases, more than others, they changed, but in all cases, we were able to achieve a model performance. In some cases, substantially better than the original. In other cases, marginally better. Autosomes was already quite good by itself. And we were able to do so with, in most cases, many fewer parameters than the original model. Again, in the case of Autosome, it was actually slightly more parameters to achieve this slightly improved performance. These models were also quite useful for interpreting sequences. So here I'm showing in silico mutagenesis of one particular yeast promoter. And you can see that these three different models have very surprisingly strong agreement across these sequences, even though they're trained completely independently and have quite different architectures, although the training data is the same in this case. But you can see that even these relatively small contributions, which most people would generally ignore, they all agree very strongly even on these sequences, which is consistent with the importance of low affinity transcription factor binding sites and regulatory sequences, which we described elsewhere. So this was supported by these neural networks. So next we wanted to see how well the DREAM optimized models generalize outside of yeast data. So we applied them to, in this case, this is a Drosophila reporter assay of, again, sequence expression pairs measured in Drosophila cells, and compared that to the state of the art, which was DeepStar. And you can see that all three DREAM models on both promoter classes outperform DeepStar pretty substantially. In this case, DREAM RNN was the best. We next applied it to human and found, again, the same thing, that all the DREAM models perform better, except in this one case where there's just not enough data to train the DREAM attention model. And so it actually performed worse than MPRANN. But again, DREAM RNN was superior. So in both these cases, these were data very much like what the models had been optimized on, these sequence expression pairs. So we next wanted to see how well it could generalize to a different kind of problem. And in this case, it's predicting ATAC-seq data, open chromatin data in human cells. And this kind of model is actually predicting the profile of insertion events of this enzyme that's used in ATAC-seq. And you can see that even here, these models perform very well. DREAM RNN is outperforming Chrome BPNet by quite a bit. and this Dream CNN, it actually should perform poorly here, but even out of the box, it's performing quite well. And the reason it should perform poorly is because of the way these convolutional stacks work. And it basically can't see the whole sequence at the same time. So if there's large distance interactions, it has no way of capturing that. So we actually expected this to perform quite poorly, and it actually performed better than we expected in this case, but we still wanted to do out of the box without any further optimization of any of these models. So now we have these state-of-the-art cis-regulatory models, and a large effort in my group right now is just applying them to all these different problems, in particular in human genetics, trying to predict variant effects and trying to design regulatory sequences. So that was one option for making continual improvement in cis-regulatory models by having these standardized benchmarks. Another way is by taking the data and models we already have and making them interoperable. So right now, the community is really faced with the problem where every time you make a model, you're going to spend a really long time benchmarking it on data sets, and the way that you benchmark it on the data sets is going to be quite different than what other people have done with the same data sets. And by the time you've published it, all these differences add up, and now your Pearson R of 0.9 is not actually comparable, even though it's the same data set, to the previous benchmark using the same data, because of all these degrees of freedom in the analysis. So here we thought it would be really nice if we could standardize things so that all models could be compared to all tasks where the model is actually suitable for that task by abstracting the communication between the models and the data sets through what is essentially an API, a standardized protocol for communication. And this is work being done by a very talented PhD student, Ishika Luthra, in the lab, and we've gotten input from a very large number of people in the field on this, and you can check out on GitHub the current state of the API here, and we would love your feedback on that. So the idea for the way this model evaluation works is that you have these containerized modules that can run in something like Docker or Apptainer, where you have the evaluator that houses the task-specific data set, a predictor that includes the model, and we also need this cell matcher module because we don't want the best model to be the best simply because it understands cell types the best. So we want this abstracted outside, and that also enables you to update this when we have a better understanding of cell types. So the way this actually works is that the evaluator contacts the predictor server with a request to predict, for instance, open chromatin in some particular cell type, HEC293 in this case, and the predictor contacts the cell matcher and says, what is this HEC293? I can predict HepG2 and HEC293FT. And so the cell matcher, using some algorithm that we still have to figure out the details of, will pick which one is the closest, send it back to the predictor. The predictor can then query the model and send back the predictions to the evaluator, where it will then evaluate the model's performance. So this approach has a number of advantages, some disadvantages too, but overall we think this is going to be a very useful approach. So like I said, the main goal was this interoperability so that all tasks can now be compared against all models. You will get a much more realistic view of real-world performance of these models because of the separation of the models and the tasks. It also should be relatively stable across platforms because everything has been containerized, and it also enables these remote servers. So for instance, if there's a prediction server that is proprietary, they don't want their model weights exposed, you could still evaluate it in this way through this API. Like I said, you can swap out the cell matcher for a different one if you're state of cell type relations changes, you can maintain this. This is very important for me because I cannot maintain a code base by myself, but if we're talking just a GitHub list of predictors and evaluators, that I feel like I could probably handle. So it should be much more maintainable in the long term. And it also enables this continual benchmarking where every year or so, to be determined, we could evaluate all the different predictors across all the different evaluation tasks. So like I said, you can check out the API in its current state at the URL below. We're looking for more people to check this out because we don't want to make something that the community is ultimately not going to use. So we're trying very hard to get this right the first time. And you can come talk to me afterward if you don't get a chance to grab that URL. All right, so part 3, we're going to talk about data leakage. So people who have looked at genomic sequences a lot have noticed that there's a lot of homology across the genome, and it's kind of obvious that this should be a problem, but we've kind of not addressed the problem adequately in the past. This was a close collaboration between Rafi in my group and Brett Kyoto from Nozomu Yachi's group. So the idea is that in some cases, there are these sequences that are present in the genome potentially many times, although it doesn't have to be many times. And in your training stage, you might have taught the model that this red and blue sequence together yield the activity of interest. But the reality could be that it's only the red that's causing the activity of interest, but the model can't tell the difference because whenever it sees the red, it also sees the blue. And we won't detect this because the same is true in the test data where we have these red and blue sequences together. So we're actually going to conclude erroneously that the model is very good because it's predicting the test data correctly, but that's primarily because the test data contains the same problems as the training data, even the same homologous sequences. So the model looks good, but actually its understanding of reality is actually flawed. And for some applications, this doesn't matter, but in the case of our wanting to, for instance, design sequences or predict variant effects, this can make a very big difference. So here's an example for attack-seek data. This is the read distribution across 2 loci, shown in the different colors, for integration sites of the TN5 transposase across the region. And these are on different chromosomes, so a model trained on one chromosome could be predicting on the other, depending on how you split up the chromosomes. You can see their alignment here. They're very close homologs. And you can tell from the overlap of the 2 data sets that they're actually very strong signals. So if the model, if all it did was memorize the one signal and regurgitate it when doing its prediction, it has no understanding of cis-regulation in that case, but it would still perform pretty well in this case. So that's a cherry-picked example, but this is generally true across the genome. So here we're using this MPRA data set from Agarwal et al. And these sequences were specifically selected because they're open chromatin regions in this K562 cell type. So it's not like we're looking at inactive parts of the genome. These are the putatively active parts of the genome. And here we're just showing the Smith-Waterman local alignment score between pairs of sequences, between the maximum from training to test sets. And this is shown for genomic sequences and also dinucleotide shuffled sequences as like a negative control here. And you can see that this homology is really common across the data set, although this is a log scale, so it's not a majority of the data set, but it is still quite a substantial issue to think about here. So one question we had was whether we could safely ignore this, because maybe, like if we're predicting variant effects, maybe it won't affect us because the variant, it's never seen the alternate allele before. So we wanted to see, okay, how often has it actually seen the alternate allele somewhere else in the genome? So we looked for these k-mers for doppelganger sequences where it's like the same k-mer, except in the middle of that k-mer, you have swapped the reference allele at the GWAS locus for the alternate allele. And you see if that alternate k-mer is elsewhere in the genome, giving the model an opportunity to cheat by looking at that. And this is actually shockingly common. So if you look at the different k-mer sizes here at 21, where there should actually be very few instances of this across the genome, over 20% of GWAS SNPs, in this case, these are posterior inclusion probability greater than 0.1. So these are like genetically probable causal SNPs. 20% of them have this doppelganger somewhere else in the genome. So predicting their variant effects, if your model has been trained on data that includes this doppelganger sequence, it might know what it does without having any understanding of why it does it. It might just have memorized it from the training data. So as kind of a positive control of this overfitting, we created this model, which we cheekily called overfit NN. It's actually not a neural network at all. It's a nearest neighbor model. So the way it works is by just taking each test sequence and looking for the closest sequences by alignment score in the training data, and then just transferring the label to the test data. So this model has absolutely no understanding of cis-regulatory logic. It's basically the maximum cheating model, where it's just finding the closest thing in the training data and returning that. And overfit NN works surprisingly well on closely related sequences. And of course, this makes sense because the sequence encodes the function of that sequence. So if the sequence is very similar, it will usually have a similar function. So here we're showing different alignment scores at the bottom, decreasing as we go to the right. And this is the Pearson R² of the quote unquote predicted versus measured expression. Same MPRA data set as before. But of course, once you get to very distantly related sequences, there's no predictive value here, as expected. If we compare this to an actual neural network, you can see the same kind of trend happening where at really high homology sequences, the performance is better and it goes down as homology decreases, suggesting that part of what these models have learned is to memorize these sequences that were in the training data. There's also this interesting dip in performance here, where these are, in this case, homologous sequences, but they're very distantly related homologous sequences. And we think that this may be like an uncanny valley here, where the model is like, it's seen something close before, so it's going to return that the prediction that was in the training data, so returning the training label, but it's actually now diverged to the point where the answer isn't the same. So it's trying to cheat, but failing. If you look at the training curves of these models, it also supports this overfitting, because if you look at the most closely related sequences here, the performance in the test data is actually even higher than the performance in the training data, suggesting that these sequences, in particular these highly similar ones, they're probably overrepresented in the training data, enabling the model to overfit them before it even has a chance to fit a lot of the other regulatory sequences in the data. So I hopefully at this point showed you that this is a problem. So how do we get rid of this, this data leakage issue? So we actually did an all-by-all pairwise alignment of the 200,000 sequences in this data set, and it took 151 CPU days, which is like, it's doable, but kind of a pain in the butt. These sequences are gonna, like these data sets, we're gonna keep getting more sequences, and they will continue to be longer as the technology develops. And since this is a runtime on the order of sequences squared times length squared, this is going to get out of hand really fast. So we wanted a scalable solution that would enable us to detect and prevent this leakage. So we turned to this locality-sensitive hashing algorithm that's commonly used in things like Amazon to recommend products to people. It's to find customers with similar buying habits, and they have such a large database of customers that they can't compare all by all. So they have to use some sort of faster algorithm to see who might be similar, and then you can check if they're really similar after that. The way it works is through this hashing process, where the goal is to actually hash similar things into the same bin. I'm not going to get into the details about how that works because it's kind of not super important for the rest of the talk, but we can discuss that later if you like. So the idea is you do this hashing process and find your closely related sequences have ended up in the same bin, and you do this a couple more times, and that way you can identify the homologous sequences. So if you take this approach, it speeds up the processing by about 100 times. And actually, it's even more scalable than that if you don't care to check whether your answer was correct, whether the things you found as probable homologs are actually homologs. Because right now, this is actually still dominated by the answer checking, the alignment of the sequences. You've narrowed it down by 100x, but you can see that the curve is actually the same shape as the original curve. It's still on the same order. You've just triaged things by 100x. And sure enough, if you apply this to actually generate chromosomal or training test splits, you have greatly mitigated the problem here. So kind of our goal with this is to make it so easy that it's hard not to do it. So if it was a pain in the ass, then everyone's like, oh, I'll just use chromosomal splits because I don't have 10 months to dedicate to this. We're hoping that if it runs in a day, people will adopt this approach and apply it to their models. And we'll hopefully be putting this on the bioRxiv in the coming month or two. So this is kind of a temporary fix. And I say temporary because we can't have models that are predicting all of genome regulation from very short sequences. And the longer the sequence gets, the bigger problem this actually is. So here I'm showing alignments between, it's actually not alignments, it's like k-mer overlap between randomly selected 200 kb sequences from different chromosomes. And you can see that the vast majority of pairs have a huge number of k-mers that overlap, indicating that they share homology compared to this random distribution, which is almost always 0 and sometimes 1 or 2. But very, very few of these 200 kb sequences have none of these k-mers in common. So essentially, at a certain scale, it is impossible to divide the genome up into training and test sets that don't have leakage. But there's another problem, in that we know, based on all of the biochemistry that's been done, that there's too many parameters for us to learn on the genome anyway. So here I'm only showing one of the parameters we need to learn to explain cis-regulation. It's the interactions between pairs of transcription factors as they bind the DNA. And based solely on the number of possible interactions they could have, there's already 220 million parameters, which is far too many to learn from the very limited sequence of the human genome. So the next approach is to improve our models by making more DNA that we can use to train the models on. So the human genome, it's been sequenced. We now have like T to T assemblies, and we can keep sequencing more people, but essentially the whole size of the genome isn't going to change that much because there's just not that much new DNA in any person. So the solution here is to create new DNA, non-genomic sequences with which we can train these models. These can include both random and designed sequences, but essentially the point here is that we are limited only by budgets and technology and how much of this we can synthesize and create and throw in cells and test. So with UC Type Halle's group, we're now working in this direction where a lot of the current effort is in developing the technology to measure synthetic sequences at different scales. So for instance, we can do short sequence reporter assays at the scale of billions of cells or much larger sequences, but in lower throughput. And the goal is to kind of measure different gene regulatory features from these sequences so that we can, for instance, learn the fine resolution of transcription factor binding and then use transfer learning to move that to larger and larger sequences so that eventually we can explain locus scale gene regulation. So I showed you that this DREAM challenge really pushed the field forward quite a bit in terms of making state-of-the-art sequence to expression models. We have this plan for a sustainable model evaluation using this API kind of approach to abstract models and tasks. I showed you that this homology-based data leakage is very prevalent in sequence-based data, and you can use hashfrag to partition it. So you should do that. And that we need more data, and we're actively trying to develop more, but I can't show you any of the results from that today. So that'll have to wait till next time. And with that, I will again acknowledge the people who were helping with this work. Most of parts 1 and 3 were done by Rafi, Brett Kyoto in part 3, and Ishika Luthra was driving the API stuff in part 2. We also had a very large number of collaborators across the globe, especially for the DREAM challenge and the API stuff. And funding, of course, our DREAM sponsors. And just as an advertisement, both my group and UBC, in particular the department I'm in, School of Biomedical Engineering, are hiring a very large number of people at the moment, 12 faculty positions, and I don't know how many we've got, but it's a lot. And we also have this new building that we're about to move into, so it's a very exciting time to join UBC. If you want, you should talk to me about it afterward. Happy to take any questions. Thanks.
Hey, Carl, great talk. And I think you made a very interesting point, an important point. As you probably know, those sequences are probably long, repetitive elements. So I have two questions related to that. They're very quick and they're related. The first one is, like, how do you know that you're actually assigning activity to these elements well? Because it's hard to map to them. And the second question is you know, I think most people would think that these long repetitive elements are going to be part of heterochromatin. So in essence, you're assigning zero activity to most of them. Is that what's going on, or do you see different activity levels for the different 20 mers that you find homologous across the genome?
Yeah, so a few things. They're not all repetitive sequences. Some of them are. You can see from here that like they must have diversity in activity, otherwise the models can't be predicting anything. And
well, it's like 0 or not 0 might give you a high Pearson, right?
Yeah, but how would it predict that? Presumably there is some signal there that was measured in the MPRA in this case. They were also derived from open chromatin. And there are many examples in the literature of even the repetitive elements developing some sort of function over evolutionary time. So some of them are
cool, and you should tell everybody what that function is. You have the data to do that now. I mean, I think people are very interested to find out the repetitive elements have activity.
Yeah. The mapping issue you also alluded to is definitely also a problem. And it's not really a problem in the MPRA data because we've kind of sucked the sequences out of the genome and tested them in this orthogonal thing. But anything like ChIP-seq or ATAC-seq, it's actually a big problem. And it will also inflate model performance that way by leaking signal across the different open chromatin or whatever it is we're talking about regions.
Thank you so much.
Thanks, Wade.
Hi. Thanks for the great talk. I'm Ekta Khurana from Weill Cornell. So the question about data leakage, this wouldn't be affecting, it's not on? Hello, is it better?
It's on.
It's better? Can everybody hear? I'll just speak loudly. So the question about data leakage would affect the performance if you're checking the same cell type. But if you try to check the effect on a different cell type, then you would see the bad performance if that was the big contributor, right?
Well, I partly agree with you, but a lot of the signals are shared across cell types. So for
Yeah,
instance, if
the
there's a
housekeeping
ubiquitous,
regions, right,
exactly,
yeah.
yeah. So in those cases, the model would be correct, but for the wrong reason. And it depends on also how you've trained these models. So for instance, if you're training on both cell types to predict both cell types, you still have that issue. But yes, there's like different ways that you can reveal that the model is now inadequate. And a lot of other groups have shown that these models are inadequate on many other tests. And this could be part of the explanation for that.
Yeah. I guess I meant the cell type specific effects, because there it wouldn't affect, and that's what we are most interested in.
Well, it could still affect that, because again, the sequence encodes for its function. So if there's like some repetitive sequence or even just a duplicated sequence that has the same cell type specific activity, this model could have memorized that.
Sure. But I think it would affect the same cell type alone.
I agree with you, yes.
One last short question. Sure, thank you for the talk. My question is again about the data leakage. This is something we've been thinking about quite a bit in the context of foundation models and so on as well. My question is it partially the reason that this kind of indirect, I would call it indirect data leakage, is particularly problematic in these kind of tasks is because you're more interested in the interpretation of the model and learning cis-regulation than the actual task itself. But if the task is there, and for example, you want to make predictions on a distant species, maybe homology information is actually something you want to take advantage of.
Yeah.
So what is that kind of trade-off, and what is your opinion on that?
No, I totally agree with you. Like in some cases, you want to know the answer. And if you want to know the answer, then it may not matter that the leakage is there. If you want to understand cis-regulatory mechanisms, then it's absolutely a huge problem because the model's understanding of reality will be wrong. If you want to understand variant effects, then it's also a very substantial problem because whatever the model has learned is incorrect. And so it can't possibly predict the effect of the variant because it doesn't understand how cis-regulation works. So it depends on the application, but in some cases, yes, it may not be an issue.